All right, to start again on mitosis, or to go through the process. So this is the entire process, as you can see from back here. So if you wanted to take a screenshot of that, you could have everything. But we're going to go through each thing. So first of all, let's talk about gap 1. So in gap 1, what we have is the original cell. Notice that it has 4 chromosomes in this case. I just drew them so that you can see them, but in reality the chromosomes would be uncoiled and loosely hanging within the nucleus, and you would only see it as like a grainy matter called chromatin. The nucleolus would also be present, and the centrioles in an animal cell. Plant cells do not have centrioles. They have microtubule organizing centers. So the first part of the cell cycle, you might remember, is called gap 1. What happens in gap 1 is that the cell doubles in size, and as it's basically getting more organelles, more molecules, everything that it would need to do the mitotic process and to create another cell. I always kind of think of this as like a plane sitting on the runway, and it's loading up the passengers, it's loading up all the food that they're gonna give you, maybe, and the drinks and so forth, and just basically kind of getting ready to go. All right, so then the next phase is called synthesis. In synthesis, what happens is the DNA is replicated. So this is just like we talked about before, or the last unit, where DNA replication is occurring, and it's creating 2 chromosomes, or 2 chromatids that are exactly alike. So right here we have 4 chromosomes, each with a sister chromatid attached to it. They're attached at a central location called the centromere, and there are then 8 chromatids in this cell, but there's only 4 chromosomes. That's just kind of the terminology that they use. The next phase is gap 2. In gap 2, the big thing that happens there is that the centrioles replicate, and the microtubules that are going to be part of the spindle start to assemble around those centrioles, which you don't usually see, but if you could see them, they'd be kind of just congregating around that spot. Now in plant cells, they just have the microtubules assembling, so basically you don't really see much, and there are no centrioles in plant cells, just the microtubule organizing centers. All right, so all of this, you might recall from the last part of the segment, is called the interphase. This is the longest part of the cell cycle. So gap 1, the cells are sitting on the runway, loading up. Synthesis is where all the passengers are replicating. No, kind of kidding. But anyway, the plane is now kind of getting ready to go. Gap 2, it is literally sitting there getting ready for takeoff. So it's all ready to go to the next phase. So here we go, taking off. We're going into prophase. Prophase is sometimes divided into 2 separate phases, early prophase and late prophase, or prophase and prometaphase, which is the next phase. So the first couple of things happen first. That is, the chromatids coil up and become visible. The second thing is that the centrioles separate, forming the spindle, and they do this because the spindle fibers are pushing between them and pushing them apart. And there are also little spindle fibers or microtubules that are hanging off the end called an aster. I always kind of think of these as like cat whiskers. They're keeping the spindle from pushing out of the cell. So it's like telling the spindle that it's near the edge of the cell. So those 2 things are happening, and meanwhile what will start to happen is the nuclear envelope will start to break down, and the nucleolus will disperse, and that leaves the chromosomes free in the cytoplasm so that they can attach to the spindle. And the spindle then will have fibers that are going from each chromatid, or each side of a pair of sister chromatids, one to one side and one to the other centrioles on the opposite ends of the poles. Here's a little kind of bigger bigger view so you can kind of see, one sister chromatid would have the spindle attached, one to one side, one to the other. And they're attached at an area called the kinetochore. It's actually a disc-like structure that allows the spindle to attach, and this disc-like structure is associated with the centromere, or the central location where the two sister chromatids are attached. All right, so then the next phase is metaphase. In metaphase, what happens is the chromatids line up on the metaphase plate, otherwise known as the equator. If you know the phrase metaphase plate, it helps you remember that it's in metaphase. All right, the next phase is anaphase. This is where the chromatids separate and move towards the poles. What is happening is the spindle is starting to disassemble, and as it disassembles, the fibers shorten, and that will pull the centromeres apart so that the chromosomes are looking like they're being dragged in jelly, which they literally kind of are, since cytoplasm is somewhat viscous. Okay, the next phase, or the last phase, is called telophase. Telophase is where the cells finally divide into two cells that are exactly like the original one, especially in terms of DNA. But the first thing that happens is the chromosomes reach the poles and uncoil. So at first you wouldn't really necessarily know that it's telophase, except for usually there's two really fairly dark blobs at either end of the cell that's dividing. Then the nuclear envelope and nucleolus reform, and then you can pretty much tell that telophase has occurred. And also at the same time, cytokinesis is also occurring. The cytokinesis sometimes is considered a separate phase of the mitotic process. Cytokinesis is the division of the cytoplasm. So the division of the nucleus was really all of the other parts that we were talking about, that is prophase, metaphase, anaphase, telophase, and the division of the cytoplasm is cytokinesis. So at the end, we have two cells that are exactly like the original. There they are at the end. There's the original. Okay, so they're exactly like the original in terms of the DNA, or the amount of DNA. But the cytoplasm has been just pinched apart, and that creates what they call a cleavage furrow. That's just kind of where they're being pinched. Eventually, they could be separate or stay attached. It depends on which type of cell is actually doing the mitosis process. Okay, so just to sum up in this particular part, this is the whole phase again. You back up here so you can kind of see everything. The whole process again. And some people remember the order of the mitotic process as pay me anytime. That is PMAT. All right, so that might help you remember. Okay, another couple of things about this. First of all, plant cells are different. So this is an animal cell. This is in your textbook. But the animal cell literally gets pinched in half. But a plant cell, because it has the cell wall, cannot do this. So it has to build a new cell wall. So what the Golgi apparatus does is the Golgi. It will make vesicles full of pectin that line up along the middle, and as they coalesce, they form what is known as the cell plate, which is basically like middle lamella between them. And then as that cell plate makes a complete line, then a new cell walls from each cell can be built, creating two distinct plant cells. Okay, so this is cell plate formation in plant cells. Another thing to consider is controls of mitosis. Controls of mitosis are occurring at various places that we know of so far, at least. Notice that one is in gap 1, so that would be right before synthesis of the chromosomes. Another one is at gap 2, that's right before the cell division starts occurring. And usually this is involved in the spindle formation. And the other one is called the M checkpoint. This is right before the cytokinesis. All right, so these are called checkpoints. We know that at least one of them is controlled via a substance, an enzyme. in this case. This is showing you the enzyme right here that is going to interact with a substance called cyclin. And as the cycle starts, what's going to happen is that cyclin, I'm sorry, do I have this upside down? I do. Sorry about that. Sorry about that. As the cycle starts over here in G1, this cyclin starts to accumulate. And as it accumulates, it's going to interact with the enzyme, and the enzyme and the cyclin will make an enzyme substrate complex that will then turn on the G2 checkpoint. That is the checkpoint that starts the actual cell division process. As cell division occurs, cyclin degrades and the enzyme is free to just hang out until cyclin then accumulates again. Okay, sorry about that.